The plan is to finish up on DNA proofreading and repair, so damaging repair. And then we're going to cover translation. And then you're going to have your second exam, which is going to be here in person. Everybody is aware of that. Yes, on Friday, on the 10th. Okay, so before I start, do you have questions? Yeah. Study guides, we have a Bell who is working on that. Yeah, so you have some study guides. Besides, remember that I put a lot of those Jove videos, those bite-sized videos, and after the entire list of Jove videos, you have those practice quizzes. So those should be, very much of study guides. Okay. Yeah. Okay, guys, I can't hear what your classmate is saying here. So let's be quiet, please. Yeah, sorry. Yes. So the stuff until next Wednesday, which is going to be on transcription, is going to be on the exam. Cool? All right. So continuing here where we had left off. We had mentioned before that you can have spontaneous mutations happening, right? And those are to your nucleotides, right? So we had here these examples. This is giving me an echo. Okay, we had here the examples of deamination, in which an amino group gets cut off, right? And then you had here this example that is showing, deamination, and I like this example because Sh2 happens, right? So from C you go to T. That's why C to T, right, happens. Okay. And then we stopped on this slide talking about tautomers. So structural isomers. Who like to have this stuff in organic chemistry? Who got driven crazy? Yay, I love organic chemistry. And you have the right hand, the left hand, and all these tautomers molecules go in your head, mirror image, non-mirror image. You don't have to remember all of that, but you should remember that you have these things that are structural isomers, so they are not mirror images, okay? And what happens here is that sometimes you have the most stable form. In this case here is a ketone type of form, right? Most of the time, the molecule likes to stay in this form, which is like over 99% of the time. But then sometimes you have this tautomeric shift in which you go from ketone to enol. And sometimes that is possible, right? You get these dynamic shifts. So when you have this type of tautomeric shifts, and it is like just before DNA replication, that is a problem because you can insert the wrong base into your newly replicated strand. So that could cause problems for your newly synthesized DNA strand. So functional isomers, here showing some more. This here you have guanine on a keto form. And when it shifts to an enol form, you see those double bonds that are shifting places, right? From ketone to enol, and that could really cause problems with hydrogen bonding when you have base pairing during DNA replication. Okay. So what is the problem here? If you have a stable form of cytosine, second. Actually, when you have here a form of cytosine that is not on the keto form and is on the enol form, and you would bind to, instead of binding to G, it will bind to adenine because it will make 2 hydrogen bonds instead of 3 hydrogen bonds. So in this case here, instead of the enol form, is showing you the keto form. So it's one of those differences in tautomeric shifts. And another example here of messed up type of hydrogen bonding, if you have your thymine that is in the enol form that is not most stable, instead of forming 2 hydrogen bonds, now you can form 3 hydrogen bonds, and instead of binding to adenine, you bind here to guanine. So that is a mismatch type of interaction. And the problem here is that if you look at the overall size of your double helix, you're not really going to see a difference in the double helix size, because here you have a pyrimidine that is getting together with a purine, so the size of your DNA is going to be about the same, right? The only problem here is that instead of T binding to A, you're going to have T bound to G. So you're going to have different levels of melting points and different interactions that you should have with certain proteins that interact with the DNA. Okay, so those are always problems that could happen, but they do not happen very often. Okay. So, all right, so if that happens during DNA replication, then you have a problem. Okay. So here is just putting into one slide what I had mentioned on tautomers. Okay. So what is going to happen here? How will you get DNA damage that leads to mutation? So let's say here that you have one of those weird base pairing, right? So you have a tautomer of adenine that got together with cytosine, and that happened during your first replication, and the problem was not detected. So now you're going to end up with one of your daughter strands that has a mutation. Okay. So this kind of mutation wouldn't really be repaired. So now for the second time, when you have DNA replication, you are going to end up with one strand, okay, that is mutated and the other one that is not mutated. After these cells undergo replication, you are going to end up with one strand that is good, one daughter strand that is good, and another daughter strand that is mutated. Okay, so when the damage is not corrected, you end up with a mutation. Okay, and these types of damages that are caused by tautomers, those are quite difficult to repair. So here, after the second replication, you end up with 4 cells, right? So 3 of those daughter cells have the correct DNA, but then one of the daughter cells will have the mutated DNA, and the cell doesn't really know that this thing was mutated. Okay. All right. Now, after the cell has undergone the mitosis and cytokinesis, and you're going to express the gene that had that mutation, you can have problems for the gene expression. So as we're going to see, you have codons that are encoded in the DNA, and when you translate the codons, sometimes these types of mutation will give you the wrong amino acid for the translation. So then you're going to have a different type of protein that is expressed in these daughter cells. Okay, so here comes your first question. Okay, so let me see here. I'll put on the confidence level. All right, the loss of an amino group from cytosine forming uracil is known as what? Losing the amino group. I was going very quickly. So the question is already telling you what is happening. Am I going to put such easy questions on the test? Probably. Okay. Does anybody need more time? Yeah? Okay. Did everybody put in your answers? Okay, I'm gonna stop it. Okay, what is your answer? C, it is deamination. All right, so here is still with the explanation that when you have these tautomeric shifts during DNA replication, that thing is not going to be detected, and the damage is not going to get repaired, and that leads to mutation. So now we have mutated DNA. So here is straight from your book showing spontaneous mutations, okay, that would need some DNA repair. And here is a table showing the types of spontaneous mutations, and they're also called endogenous DNA lesions that happen every day. That means in 24 hours, you have purines that are flying off your DNA, like how many? Well, about 18,000 of them a day, okay? Pyrimidines, they also can fly off, but not to the same extent of purines, because purines are just bigger, they'll fall off easier. And you have all these different types of DNA damage that can really happen just because you are alive. So that's just a FYI type of chart. Okay. All right. Now we have different types here, still other types of damages. You can have, instead of putting the correct purine, you can have a different purine that is inserted, and we can see that can happen because of tautomeric shift base pairing. Okay. When you exchange one purine for another, you call that type of DNA damage, you call that transition. Okay, so you have one purine that is shifted to another purine during the DNA replication. So you say you have a transition mutation. Now, instead of having a purine that shifts to another purine, if you have a purine that shifts with a pyrimidine, you call that a transversion mutation. Okay. And the same thing like the other way around. If you have a pyrimidine that is shifting with a purine, you call that transversion. If you have one pyrimidine that shifts with another pyrimidine, you call that transition. Okay, so if it is the same type of base, you call transition. If it is different type of base, you call transversion. So here are more causes for DNA damage. Right? We have looked briefly, we're going to look into more details about UV radiation. And here we show oxygen, reactive oxygen species type of damage. First, how do you get reactive oxygen species? Well, if you have a peroxide molecule, for example, and you break the molecule, sometimes you can have a free electron that goes with one oxygen and a free electron that goes with the other oxygen, because those two atoms have the same type of electronegativity. So one of them is not going to hold more to the electron than the other oxygen. So you might have this free radical that shows up. And free radicals are quite dangerous. They tend to react with anything that they close to. So if they hit whatever they hit, they're going to react with it. It could be fat or carbohydrates, proteins, or your DNA. Okay, so anytime you have free radicals, you can have some type of damage, and if it hits the DNA, then you can have DNA damage. And actually, free radical formation is a major cause for DNA mutation. Okay? And what that leads to could be transformed cells. Right? So you can have some cancer cells that could show up. And another hypothesis that is given to action of free radicals is that it could accelerate aging. And aging is actually quite complex. And I just saw an article today that was kind of turning my head upside down, because I always thought that aging comes from the inside. Like, your cells are not getting younger, some cells are going to stop dividing, some cells are going to undergo senescence, that means they just stay quiet. So I really thought that, formation of free radicals will come from the inside to the outside. And today I just saw this article saying that aging has a lot to do with changes from your skin that go from the outside to the inside. I'm like, what in the world? You never know what you're going to learn. And it was making quite a lot of sense. So now I will definitely start paying more attention to skin care. Yeah, I'm not going to go off on tangents here, but it does make sense. Have you noticed that people who suffer from severe depression, they tend not to shower? Now it kind of makes sense, and they tend to look older after a period of time, older than somebody who was the same age. After reading this article today, I'm like, oh, yeah, actually it would make sense. But anyway, let's come back here to free radicals. Free radicals. That's one type of DNA damage. Another thing that can cause DNA damage or DNA mutation, okay, this is more for the mutation, is presence of transposons. Okay, so those are also called jumping genes. So those are pieces of DNA that can be moved from one site to a different site. And this happens randomly, right? It's not like we have pieces of DNA flying from one side to another side of your whole chromosome right now all the time. That would be a huge mess. But it happens over time, and mainly it was a mechanism, or it is a mechanism used by retroviruses. Okay, so viruses such as HIV, okay, they have a protein like this that is called a transposase. Okay, where is the transposase? Here. Okay, so this is an enzyme called transposase. The transposase recognizes a stretch of DNA that is usually a palindrome. Have you heard of palindromes? Yeah, palindromes, back and forth. Okay, race car, palindrome. Okay, so transposase goes there, recognizes a palindrome, and it cuts it just like a restriction enzyme in bacteria that we briefly looked before. Okay, so after it cuts, it inserts a piece of the DNA that was actually a cDNA, or is a copy DNA that came from the virus, right? So it inserts a piece of that DNA, and then it allows polymerase to do the connecting part of it. So now you end up with an extra piece of DNA that wasn't there and wasn't even your own, but now it becomes your own, and you're never going to get rid of it. Although there are some researchers showing that you can actually cut HIV DNA out of some cells, but that's a different story. So that has to do with the latest scientific advancements and goes beyond the scope here. All right. So basically, transposons, they go in, and that's it. You're stuck with it for the rest of your life. Another cause of mutation could be, like I mentioned before, certain bases that look like the regular base. They behave like the regular base, but they are not. So you call those analogs, base analogs. Okay. The problem with those base analogs is that they are going to base pair strangely with whatever they encounter, and if it is doing replication, then you have a problem. And then you have these guys here. Those are alkylating agents. So when you mention an alkyl group, that has to do with a methyl group or ethyl group, small type of hydrocarbon. Guys, I can't talk with a chat on the back of my head. So you get here a type of methyl group or ethyl group. Those are your alkyl groups that get stuck on your base. So here you have an heterogeneous base, and you have a functional group right here that can receive an alkyl group. So now you have a base that is part of your nucleotide, And this thing is totally useless because it's going to damage the DNA, and this cell is probably going to die because it cannot correct for this DNA damage. So one example of an alkylating agent that is very much well known is mustard gas. So here is the molecule of mustard gas. And this was actually used during World War I as a chemical weapon. Okay? So it was after World War I, this type of mustard gas or alkylating agents or chemical weapons were banned by the Geneva Convention, but that doesn't mean that they're not getting used still, these days. What does it cause? You have severe burns that develop. It will be the same case of like extreme high radiation levels. It causes blindness, and it causes tumors. So this is a slow and painful death. So for some reason, politicians think that a quick death is better than a slow, painful death. I mean, quick death caused by war, and let me not get into political thoughts. So I think every kind of war is wrong. It doesn't matter how you kill your opponent. I think killing somebody is just wrong. But that's my opinion. It's already so tough to deal with life, right? So coming back here, things that can cause problem in the DNA. Right? You have intercalating agents. So these are not base analogs, but they are bulky things that can stick themselves in the middle of a DNA double helix. Now, examples of this, which I don't have the molecular picture here, example of this is ethidium bromide. Anybody heard of ethidium bromide? No? Yeah, one person. Yeah. Did you run DNA gels in the lab? Oh, genetics class. Okay. Did anybody use ethidium bromide in the lab? Yeah. And first thing that you hear about it is carcinogenic. Right. But until, I would like to say recently, but it's actually, I don't know, 20 years ago? Recent enough. Okay. It was very common in the lab. You just order it from like Sigma or something, and you put like half a microliter of this in your gel preparation. And then you're going to run a DNA gel, and then you get beautiful bands because they shine. Like you put UV light and they glow. Okay. And it's beautiful. You find your piece of DNA, the correct size, you cut it out, you do your cloning, everything is good. But it happens that this thing is carcinogenic because it can intercalate in the DNA and can cause mutations. So nowadays they have this substitute for ethidium bromide. There is some kind of orange something that I never remember. Oh, here we go, acrylic orange. Okay, and this thing is not as carcinogenic and is safer to use in the lab for routine DNA gel type of things. The problem with this is that it doesn't shine as well as ethidium bromide. So instead of using like half a microliter, you gotta put, I don't know, 5 microliters of this thing to try to look at your DNA. It's kind of a pain in the neck. But anyway, things we do in the lab. Okay. And that was intercalating agent. So here's another thing that can cause mutation. Or actually, we're talking about damage. So another thing that can cause a type of damage is UV light. So this is ultraviolet light. Now, UV light, the length of the UV light is around 260 nanometers. And that is exactly the size that would be in synchronicity with your DNA molecule. Okay? Because that has to do with the electron cloud of double bond, intercalating double bonds. Okay, so what happens here? When you shine this UV light on the DNA, you can excite this electron cloud, and this type of electron excitation can jump to another electron cloud that is also excited. So what happens here is that if you have these types of electron clouds close to each other, in the case of two thymines that are close to each other, you can end up with a cyclobutane ring. So this type of interaction is actually forming two covalent bonds between two of those nitrogenous bases. And this is really bad because it causes a kink in the DNA, in one of the DNA strands. And when the cell undergoes synthesis, DNA synthesis, so polymerization, the DNA polymerase gets stuck in there. It doesn't know what to do with this bulky thing of DNA. So it stops the replication process. So this thing is bad, okay? So this type of DNA of thymine dimers, they have to be corrected, because if they're not corrected, they're going to cause mutations. So a lot of those happen throughout the day. So here is from your book showing what happens when you have UV radiation and what happens here to the molecular arrangement of those covalent bonds that can form with those thymine dimers. Okay. All right, so here is another question, and everybody should get that question correct because I just gave you the answer. In the formation of cyclobutane, pyrimidine dimer, which of the following bonds are involved? Okay, does anyone need more time? No? I'm going to stop it. What is your answer? C, that is your covalent bond. All right, so other things that can cause mutation. Here you have high energy radiation. Okay. What kind of high energy? You can have X-rays or gamma rays or even cosmic rays coming from different planets or suns or stars. Okay. Now, how are those high energy radiation rays going to give you problems? They can cause free radical formation, and then the free radical will cause DNA damage. Or they can directly interact with the DNA and break the strands. Then you can have, a double whammy type of thing. You have free radical formation, DNA damage, DNA breakage, right? So this is very detrimental for proper cellular function. Now, when you talk about ionizing radiation, you should think that is a form of electromagnetic frequency, right? And here is just a chart comparing what are the types of radiations that you're exposed to. So here you have, for example, frequencies that are not very high, right? So very, very large waves. You have here a type of computer waves, radio waves, so very large waves. Cell phone waves, and then you have microwaves, and that starts to get into the bad side of things. Remote control waves, I never thought of that. Okay. And then you have this to this side of ionizing radiation. Okay, so the frequency of ionizing radiation is from 10 to the 14 and up. So that means the wavelength is very, very, very small. Okay, if the frequency is high, the wavelength is very, very small. So here you have visible light, and visible light is going to also bring in UV light. So you have the whole rainbow here. Okay, you have UV radiation that comes over here, and then you have X-rays, and then gamma rays, and worse. So those are your ionizing radiation. So here I have this picture showing accidents that happen involving radiation. One that was the biggest accident that I could remember was 1986, and that was a Chernobyl accident. Have you heard of this? Yeah? Most of you were not born at the time, but Chernobyl was a big. You were born? Yeah, me too. Okay. You were there? Oh, your mom was there. Oh, wow. Well, I hope she went out of there very quickly. I hope she got out of there really quickly. Wow, that is scary. Yeah, so yeah, so you know everything about Chernobyl. Yeah, so even nowadays, the area is still high in radioactivity, and most of the area is totally deserted. But they do have like some, I don't know, farm animals. So you get radioactive cows, radioactive milk, and not fun. Okay, not fun stuff. So huge problems with the Chernobyl accident. And more recently, we had in 1987 this accident in Brazil. Anybody heard of that one? There were not as many people who died, and it was not actually involving a nuclear reactor. What happened here was that this site used to be a hospital, and they used to have radioactive waste because hospitals actually run tests and they use some radioactive materials, and then they put the material in this very isolated type of container, and this hospital was not worried about what to do with the radioactive waste. And the hospital went out of business, it got demolished, and the waste stayed there, and nobody took care of it. Yeah, sometimes, Brazilian government is kind of a pain in the neck, I can tell you firsthand, but I know. So, what happened? There were some people that decided to go and do some scavenging hunt for things that they could save out of these ruins. And then they found these capsules with a symbol in there that they had no idea what it meant. Of course, it was a radioactivity type of symbol. So they broke these capsules open and they found this shiny powder that was glowing in the dark, And what did they do? They took it home, of course, and they gave it to their kids. They gave it as a gift. Look, it's shining in the dark. So the kids are like painting their faces. Yeah. And they were painting stuff on their walls. And the whole village got contaminated. You have 60 people who died and 6,000 people who got radiation contamination. Yeah, kind of dangerous. So this was recent. And then even more recently, we had the Fukushima disaster, but that wasn't as bad. So this was 2011, it was because of the tsunami. So this was in Japan. They did have this type of radiation leakage, but nothing compared to what happened to Chernobyl. All right, so types of DNA mutation. So this stuff is fun. Sometimes you have a mutation, and the mutation is not going to change anything. So here you see, for example, you have your original DNA with CCG, and then you get a mutation that changes the C to a U. And this thing here with the new mutation is coding for the same amino acid as the original DNA sequence. Okay, so this is a type of silent mutation. It's not going to make any change. Okay, usually that type of silent mutation happens if the mutation is happening at the third nucleotide of your codon. So your third nucleotide, as we'll see next, that is your wobble base. So usually if you get a mutation in the wobble base, you don't have to worry about overall big mutations because that's a bit more lenient. Okay? So this first type of mutation is your silent mutation. The next type of mutation is when you get a wrong amino acid that can come out of that mutation. Okay. So in this case here, you change your first amino acid. So instead of a G, you have A now. So your codon here will code for serine instead of glycine. Remember, glycine is your very small amino acid, right? And now serine is kind of bulky and has a hydroxyl group. they can interact with other things that glycine could not. Okay, so now we're going to change your amino acid type of mutation. Okay, so that is not a silence mutation. That is called a missense mutation. Some other times you can change the amino acid, you can change the codon, but the type of amino acid is basically with the same type of properties as your original amino acid. So you also call that a missense mutation, but in that case, you would not change the overall properties of amino acid. So in that case, you would have a neutral missense mutation. Okay? So two types of missense mutation. You have neutral or you have plain missense mutation. Okay. Then the third type, second type, and then the third type of mutation you can have is when by changing one of the nucleotides, instead of getting a codon for some amino acid, you end up with a stop codon. So when you have a stop codon, you're going to stop translation. Now you end up without amino acids there, and not only without amino acids here, without amino acids everywhere else for whatever the gene was coding for. So you end up with a truncated protein, which is basically useless. Okay, so this is called your nonsense mutation, when you end up with this stop codon. All right. So here is easy representation of what those mutations mean. Okay, so in case of okay, we'll see the other mutations. So let's see now in case of insertions or deletions of nucleotides. Okay, so here we say in case you have insertion, you can stick one nucleotide in the DNA where it did not belong there. Okay, so what happens now? At first, you had something that would read like with 3 nucleotides codes, right? So at first, you have this sentence that says the dog with the man. So if you insert something there, when you look at the DNA, you can still see the codons, but when you do the transcription and translation, your translation is going to translate to something that you cannot identify because you're going to have a frame shift on your reading frame for translation. Okay, so when you have insertion, in this case here, you always have to read everything on 3 base pairs, right, on 3 nucleotides, right? So in this case, you would have a translation that would read like the dog and so that's not going to give you the proper protein, right? You were looking for the dog with the man, and you got something that had Emma in it, so it's not the correct end product. Okay. On the other hand, if you have a deletion of a nucleotide, in this case, let's delete the G out of the dog, right? After you go through translation, you end up with a protein that says the what? Dog eat him, and still is a wrong protein, right? You almost had the dog here, but the rest of the protein is totally wrong. So either deletions or insertions of nucleotides can cause frame shifting. Okay. And the frequency of those frame shifting, you can think in your head, like if you add 1 nucleotide, yeah, you're going to have a frame shift. You add 2 nucleotides, still frame shift. You add 3 nucleotides, now you're going to have a whole codon that get inserted in there, so you're going to have an extra amino acid in your protein, but then everything goes back to frame. Okay, so we're going to have a mutated protein that has 1 more amino acid that it's supposed to have, and also the other way around. So here's just a picture showing, substitutions or deletions or insertions. I thought that was a nice visual to have there. Looking at different bigger types of mutation. Okay, so what we have seen with insertion, deletion of single nucleotides or substitutions, those are looking at genes, as small sequences of genes. Then you have type of mutations that have to do with like big chunks of DNA. So examples of that are when you have addition of trinucleotide repeats at the end of chromosomes. So we have seen that the telomeres have a lot of repeat sequences, right? Because that's like the garbage DNA, the buffer type of DNA that allows the cells to undergo cell division, and you know, the Okazaki fragment side, and the piece does not get replicated. That's a buffer region at the end of the chromosomes. But then sometimes you have certain problems that arise because you might have too much addition of nucleotides at the end of the chromosomes, in particular some chromosomes, and that causes overall problems to cellular function. So here we have what we call expansion of trinucleotide repeats. Okay, so everybody has trinucleotide repeats, but sometimes they can be expanded to sizes that are not normal. Okay, and usually when you see abnormal type of increases in trinucleotide repeat, you're going to have, instead of like 20 or 30 repeats, you're going to have like more than 200 copies of those trinucleotides. So that that can cause problems for cellular expression. Nobody really knows why that happens, but one hypothesis is that you have a polymerase, the DNA polymerase, that is not doing a good job. So remember, when the DNA is doing the polymerization, it reads the template strand and it makes the new strand. But sometimes with those trinucleotide repeats, the polymerase can jump back to a place that it already replicated and keep on replicating and kind of get stuck in that. We call that DNA slippage. And what ends up happening is that you might have these trinucleotide repeats that keep on repeating, and then one part of the DNA can form this type of hairpin loop. So everything that is involved at the end of this chromosome is going to be very much messed up. So this is an example of what happens with Fragile X syndrome. Okay? And you see here the example of Fragile X syndrome, that the X chromosome has these structures at the telomeres that they're not supposed to be there. Okay? Another example of polymerase slippage is seen in Huntington's disease. And you can imagine Fragile X is inherited, but it is recessive from what I remember. But Huntington's disease is a dominant type of mutation. So that means if you inherit only one of the alleles from one of the parents, you are going to have the development, the phenotype development of the disease. Now, Huntington's is particularly detrimental because it doesn't show up until a certain age, usually after middle age. So only after 50, 60, or 70 years old that you're going to start showing symptoms of Huntington's. And that is a big problem because by that time you already had like a whole life behind you, and you probably already had kids, and you can as well pass the genes down the line, right? Nobody's going to find out until the person gets to be middle-aged. So this is a kind of a serious situation. Now, not only this disease is dominant, sometimes we see what phenomenon that we call anticipation. That means every generation that is inheriting the mutation, that's inheriting the disease, they tend to show the symptoms at an earlier and earlier age. Okay, so let's say if the grandfather started to show the symptoms at 80 years old, then the father would start around 70, and then the son would start around 60, and the grandchild would start around 50. So you see this type of starting symptoms, age of onset, to be earlier and earlier as the generations progress. So that is called anticipation. So, well, Fragile X, it already has such a huge chunk of DNA that is replicated that I don't think that that is the case. I think it's more with Huntington's. Because also if you look at the symptoms of Fragile X, that is like one of the main causes of delayed learning or mental retardation. And usually it shows up very soon, very early in life. It's not like you're going to wait for your whole life for the symptoms to show up. So usually it usually happens in boys because they have only one of the X chromosomes, and it's recessive. So if the girl has the gene, most likely she's not going to express it. You have a problem of just going down the line in case of recessive, having a boy. So yeah, with Fragile X is different. It shows up right away. But yeah, with Huntington's is the other situation. And actually, there was another study that came out a few weeks ago showing that Huntington's onset of symptoms could be delayed for, I think, about 75% of the time that they would expect based on gene therapy. I didn't read the article yet, but that's very promising. It's very, gives some kind of hope to people who already have the disease. So this is a FYI for you to know what types of syndromes are associated with nucleotide repeat sequences. Right, so here you have, of course, the one that you have to know is Fragile X and Huntington's, okay? And then you have all these other types of diseases that you can read in case you want to figure out more about this type of problems. So here, more types of mutations that you can have is when you have either different numbers of chromosomes or different pieces of chromosomes that get shuffled from one side to another. Okay, so you can have like a different number, is not showing here, but here showing a different piece of the chromosome that got shifted from one side to another, from one chromosome to another chromosome. So let's see here. The term aneuploidy, it means that you are either going to gain or lose one chromosome or more of chromosomes. So we'll see examples like very soon. Okay, so let's see here. Example of monosomy, it means that you got rid of one of the chromosomes. So instead of having 2n number for each chromosome, one of the chromosomes, you're going to have only one of them. Okay? So examples of monosomy is when you have here a Turner syndrome, that the woman is going to have, instead of 2 X's, the woman has only 1 X, and the O here is not a chromosome, it's just to show you that there's nothing there. Okay? So XO, that is Turner syndrome. You could think that, well, the woman started with 2 X chromosomes in the first place, and one of the X chromosomes gets silenced, right? That's a bar body. That's one of the epigenetic mechanisms. So why is it that XO is a big problem? Well, the chromosome that usually gets silenced is not that it's totally quiet. It has some function. Okay? So when the other X chromosome is totally absent, you do have developmental issues for this woman. So usually they have certain characteristics. They're like short stature, and they do not develop very good secondary sexual characteristics. You have a lot of issues that are involved with this type of mutation. Another type of aneuploidy is trisomy. So in this case, you're going to increase the number of chromosomes. So here I have an example here, XXY, that's Klinefelter syndrome, in which you have a man that has 2 X chromosomes instead of only 1. Now, every time you see abnormalities of sex chromosomes, if you have a Y, it's always going to be a male. Okay? Now, if you don't have a Y and you have a lot of X's, that is, of course, always going to be a female, well, most of the time. Okay. Besides Klinefelter syndrome, you can also have like trisomy 21, that's Down syndrome. That's the most common form of trisomy or aneuploidy. And then if you have other examples here in case of tetrasomy, that means you have 2 extra chromosomes of the same type. So instead of having 2N, you have 4N. Okay, so it's a lot of extra chromosomes there. Also not very common, not common at all. Then you have this other form of big chromosome type of mutation that is called polyploidy. And this means that instead of getting one extra chromosome here or one extra chromosome there, you get a whole set of extra chromosomes. So this is not viable for animals, but is very much viable for plants. Okay? And actually it's kind of useful for certain plants. Like for example, when you go to the supermarket and you go to buy strawberries, who likes strawberries? Yeah. Have you seen some strawberries that they have they're more like the size of apples instead of strawberries, and they're really juicy and really sweet, you know? Those have abnormal number of chromosomes. They have extra sets of chromosomes in them. Yeah.
Does it say?
Polyploidy? Yeah. Sorry. This thing here that is always in the middle. I was trying to redo my slides so that I will not use this extra little space in the board, just in case. So with polyploidy, so you can get some fruits that are, kind of huge. Right? So that's accommodated in plants, but you will never see this with animals. Okay. So you can have diploidy, in which you have a whole set. Triploidy, they have two sets of extra sets of chromosomes. So here is an example of aneuploidy. And this is chromosome 21 that gets one extra chromosome. So it's a trisomy 21. Okay. And we see that happening in humans, yes. So here is your chromosome 21, which is very, very small. It's not the smallest chromosome. The smallest chromosome is 22. Again, when you look at karyotype, you'll see that the big chromosomes, those are the small numbers. So the big one is like number 1, and then number 2, number 3, and they decrease in size as the numbering goes up. So when you get to chromosome 21, it could happen that instead of inheriting one from mom and one from dad, you inherit 2 from one of the parents, and it's usually from the mother. Okay, so there is this correlation between the advanced age of the mother and the incidence of trisomy 21. Okay, so we see here that the borderline is close to 35 years old. When you get to 40 or 45 years old, the incidence goes up, and this is per 1,000 births. So you see that you can have like 10 per 1,000 births if the mother is around age 40. And that is nowadays an issue because a lot of us women tend to postpone having kids because we have school, we have college, we have graduate school, we have career to deal with. And then you put kids in the middle and it gets more complicated. Believe me, it gets a lot more complicated. Because the mom is usually the one to take care of the kids, saying all the time, So when you guys get married, make sure you have equal opportunity taking care of your kids, especially at night. Yeah, not like you're gonna breastfeed, but you can always pump and put the milk in the fridge so dad can go and breastfeed this kid that is screaming 5 times in the middle of the night. But life gets complicated. I would advise ladies not to have your kids during your graduate school if you're planning to go to graduate school. That is not very much compatible with life. Believe me, I know. I had my son when I was in my first year of PhD. Not fun. Okay, I survived. It's not impossible. And then what happened? 5 women in the department, they saw that I was surviving with a baby and they decided to get pregnant too. The chair was not very happy. I said it was not my fault. I said there was something wrong with the water. Anyway, don't. Unless you have family support to take care of your kid when you're studying and working. All right, so different. Okay, don't delay too long to have kids either, because then by the time you're 50, your kid's going to be 10 years old and you don't have enough energy to run after them. Okay, difficult. All problems apply. So, okay, coming back to other mutations, right? You have some type of chunks deletions in chromosomes. And in this case, this is a very strange mutation. This is a mutation that happens in chromosome number 7, so that's a big chromosome. And you lose a whole chunk of chromosome that is important for expression of a protein that is called elastin. That's a protein that is important for skin and some tissues that have to expand and contract, like your lungs, right? And then what happens? What are the phenotypes of this type of mutation? Well, you end up with a whole bunch of phenotypes and you put it together in the syndrome that is called Williams syndrome. So what happens to this kid? First of all, these people are very friendly, they're really happy, they love music, and they're party animals. They love a party, okay? And actually they have camps for Williams syndrome children and adults. And of course the camps, they have a huge, kind of exposure to music of all different types because they have a very, very good ear for music. There are some Williams people that they can just hear the music the first time and sit down and start playing the piano like they already know the music like by heart. It's amazing how they develop this musical ability, you know? But on the other hand, their cognition and overall social skills, they're very much delayed. So they don't really interact for a long time. Yeah, they're party animals. They go, they go and party. But if you try to talk to them, they don't really have a conversation. They have nothing to say. So let's party. Okay, who needs to say anything when you're partying anyway? So, and listening to music and dancing. So there are some limitations. So that's Williams syndrome. And then you have this other type of big chunk type of chromosome mutation, Cri-du-Chat. Okay. Look at this little girl. You see that she has some facial features that are not considered normal. And most of the time, this type of chromosome 5 deletion goes with microcephaly. So the size of the head, of course also the size of the brain, is smaller than usual. Okay? And the reason why this syndrome is called Cri-du-Chat, okay, so it's a piece of the chromosome 5 that got cut off. The reason is that genes that were present here, they are very important for development of the throat, of the larynx. So without those genes, the larynx does not develop in a proper way, and the voice of these babies end up to be kind of sounding like a cat. I mean, newborn babies, they already sound like strange little creatures, but in case of Cri-du-Chat, they would really sound like a crying cat. Okay, so that's that's like the first telltale sign that you have some issues with this type of mutation. And this is not inherited, right? Because of course the person that has the syndrome has a lot of developmental issues. Usually they do not have children of their own, right? So it's not really inherited. And this type of mutation happens just by chance. This is what you call de novo or sporadic type of mutation, right? Okay. So other features or expressions is besides microcephaly, you also have low birth weight, also cardiac issues, the same way that we have throat issues. And what else here? Oh yeah, widely set eyes. So you see like the separation of the eyes is larger than normal. Okay, so here, oh, I didn't mention this disease yet. All right, so here you gotta go by elimination. Let's see what happens, right? Which disease is associated with defective nucleotide excision repair that has to do with sensitivity to UV light, and I'll tell you something else. This has to do with the cyclobutane ring of thymidines. Okay? So go by elimination. Consult your friends who probably have seen the PowerPoint before class. Oh, not as bad. If you see an option that is something I did not mention, it's probably that's the wrong answer. Okay? Okay, so most people got it correct. Can I stop it? Anybody need more time? No, everybody go? Okay, what's your answer? Hey, yeah, there's enodermal pigmentosum. So I'm glad that most people either looked ahead or guessed correctly. All right, that's what we're going to get into next, right? This point should be after. I have to shift it for next classes. So what happens? We have seen the damage. So now let's see the repair, okay? So we are going to look at different types of repair, and keep in mind that some of these repair mechanisms, they can have redundant function. It means that the mechanism is so important that if something does not get corrected by one thing, it will be corrected by another thing, because if you don't correct it, that is a big problem, okay? So we're going to look at the ways that you can correct when you have a small type of mutation, like when you have some bases that got some alkyl groups, or if you have some analog bases that got stuck in the DNA. So small type of mutations. Then you're going to see at repairs in case you have those dimers, okay, or interpolating agents. Then you're going to look at single-stranded breaks and then double-stranded breaks. So you're going to know the types of repairs that are mainly involved with each of those types of mutations. It's going to have a little toolkit for repair. So first thing that you're going to think here is, how is it that you're going to detect the mutation? How is it that you're going to take out the mutated piece? And then what is going to jump into function to repair the DNA that ends up without a piece? Okay. And also there are some diseases that are very much associated with specific type of mutation repair that is not repairing anything. It's dysfunctional mutation repair. So here are the types of repair mechanisms that you absolutely have to know, okay? So direct repair, this is mainly important for bacteria, not for large animals or us, okay? Direct repair, nucleotide excision repair, there, base excision repair, there, not beer, right? Mismatch repair, homologous recombination, and non-homologous end joining. I know these are just words, they're going to make sense in a minute. So here, when you look at those thymidine dimers, okay, you see that bulky DNA, right? Now, what is it that causes the type of damage? UV light, right? So now it will make sense that you could have a direct repair of this problem by using an enzyme that gets also activated by UV light. So you're exposing yourself to the cause of the damage, but the UV light is also activating a protein that can go and repair the damage that UV is doing. Okay, UV is activating the protein too. Okay, so it's activating the problem and the solution at the same time. Okay, then the name of that enzyme is photolyase. So the name is telling you is activated by photons of light, and is going to cut, lyase is going to cut that cyclobutyl ring, and is going to repair directly your thymine bases. Okay, so here is what happens. Your light was shining in here. Do I have us? Okay, so in case here of animals, we do not have the direct enzyme repair, or we do not have one that we have detected so far. Okay, so you have this type of mechanism that is called excision repair. Okay, so what you're going to do, you're going to cut a piece of the DNA that got mutated, end up with a piece that is only one strand of your DNA, and then polymerase is going to go there and it's going to repair the gap. Okay, so this is type of excision repairs. You have two types of excision repairs. So main idea of this is that you have a nuclease enzyme, which is actually a endonuclease, because this thing is in the middle of your double helix, it's not at the ends. You have endonuclease that goes in there, cuts the piece out, and then DNA polymerase fills in the gap, and then DNA ligase is going to seal the gap, just like we have seen before. Okay? Two types of excision repair. You have base excision repair, and we have a nucleotide excision repair. Okay? So here, two types. And here is when I finally talk about xenothermal pigmentosum. Okay? So this is a type of repair that is very important to undo those timing dimers. So let's say you go out in the sun and you get all these timing dimers, and then you put this repair mechanism into action. Okay? Timing dimers is the most common. You can also have other problems that have to do with photo products, okay? But I usually just stick to timing dimers. Okay. So what is going to happen here? If you don't have this repair mechanism, you end up with a xenothermal pigmentosum. Okay, we're going to see more what that means. So how is this happening? You have your excision enzymes that get there, and they cut out not only the part that was damaged, but also a part far away from the damaged site. And this is like good nucleotide that gets pulled out also. Once this piece got cut out, then DNA polymerase goes there and fills the gap, and then the ligase does the job, and you end up with a fresh DNA double helix. So here's the case with xenothermal pigmentosum. And the main cause for this is defect on your nucleotide excision repair mechanism. When you have this type of syndrome, you're going to have a lot of problems with the skin, because your skin cells cannot repair the damage. So you cannot be exposed to UV light. You cannot go out in the sun at all, basically, unless it's like very, very dim light, very early in the morning or very late in the evening when it's almost dark, okay? What happens? You get freckling, you get ulcerations, ulcers, and you get skin cancer very, very quickly, okay? Very serious problem. Now, we have seen that exposure just going out in the sun can cause a lot of timing dimers throughout the day in everybody, like over 1,000 of those a day. And your nucleotide excision repair jumps in and fixes up the issues. Okay, so here with the xenothermal pigmentosum, that doesn't happen. So actually, I had seen one case of xenothermal pigmentosum at Hollywood Beach when I used to have more energy in the mornings, which is not the case right now. I used to wake up like 4:30 in the morning and go to the beach, and go for a run and do my 5K early before kids were born. So yeah. So and then at that time, just before the sun was up, there was this guy that was riding his bike at the Broadwalk, and he was totally covered. Like he had this bodysuit that had only a little bit of a window that he could see. Like the whole body was covered, hands, everything. So I'm like, oh, that's a case of xenothermal pigmentosum for sure, because he would only show up at that time, and not even in the evenings, just early in the morning. And so there is a certain level of UV exposure that the person can be exposed to, but it's not much. Actually, they have these camps for kids who have this type of syndrome. It's called Sundown Camp. Heard of it? Yeah, a Sundown Camp. They have camps during the summer that is only at night. So it's for these kids that really want to go out in the sun and have a normal, not in the sun, normal. But the whole family has to adapt to the needs of these children. Oh, here is that example again. Oh, okay, you already got this one right, so forget about it.